Hello everybody. In the last class we have studied about the different types of bacteria and their classification on the basis of number and arrangement of flagella. Today we will be going to discuss about the structure of bacteria. Bacterial cell structure. So as you all know that bacteria is a prokaryotic unicellular organism. Bacteria is prokaryotic unicellular organism. So we would be studying its structure one by one. Bacteria has a outer layer known as the capsule. So we'll start from the outside like this. This is the capsule of the bacteria. First structure we are going to discuss is the Glycocalyx. Glycocalyx. The capsule is made up of this Glycocalyx. Capsule is made up of Glycocalyx. This glycocalyx is further composed of complex polysaccharide or protein. Complex polysaccharide or protein. So capsule is the outermost layer of a bacterial cell. One more layer is there in some type of bacteria, that is known as a slime layer. Slime layer. We would be discussing this later on. First of all, we have to discuss about the capsule. What is capsule? It is defined capsule. Capsule is the outside of cell, present outside of the cell. It prevents the cell, it prevents the bacteria from phagocytosis. Bacteria from phagocytosis and is responsible for the pathogenicity of the bacteria. Responsible for the pathogenicity of bacteria. What is pathogenicity? It is the disease causing ability of a bacteria. All the pathogens, all the microorganisms that are pathogens have a capsule around their cell wall. The examples of bacteria having capsule are Streptococcus pneumonia and Haemophilus influenza. Let us see the examples. Examples are Streptococcus pneumoniae and Haemophilus influenza. So what is glycocalyx? It is the outer layer present outside the cell wall. It prevents the bacteria from phagocytosis. It is responsible for the pathogenicity of bacteria. The examples of the bacteria having capsule are Streptococcus pneumonia and Haemophilus influenza. Now the second layer, one more layer is there, that is the slime layer. Slime layer. It is also made up of glycocalyx. Glycocalyx. As we have studied about capsule, this capsule is tightly bounded around the cell wall. It is tightly bounded. Tightly bounded layer. But slime layer, it is loosely bounded layer. Loosely bounded layer. And what is the function of this layer? It prevents dehydration, and nutrient loss, and it helps in trapping nutrients. So have we studied the outer structures? This is the glycocalyx, and the glycocalyx can be a capsule or a slime layer. Capsule is made up of glycocalyx and slime layer is also made up of Glycocalyx, but the difference is that capsule is tightly bounded while a slime layer is loosely bounded, and a slime layer helps to prevent dehydration and nutrient loss and help in trapping of the nutrients. Next structure of a bacterial cell is the cell wall. It is inside the capsule. So this is the cell wall and this is the capsule. So next structure of bacteria we are going to study is cell wall. The cell wall the cell wall of plants is made up of cellulose and that of fungi is made up of chitin. But in bacteria the cell wall is made up of peptidoglycan. The cell wall of bacteria is made up of peptidoglycan. As the name suggests, it has a peptide portion it is made up of amino acids, and this glycan portion says that it has sugar portion also, sugar. There are two types of sugar present in a cell wall of bacteria. These are N-acetyl glucosamine and N-acetyl NS. One of the sugar is N-acetyl glucosamine and the another one is N-acetyl muramic acid. And these both sugars are bonded by a bond known as beta-1,4 glycosidic bond. These two sugar are bonded by a bond known as beta-1,4 glycosidic bond. So on the basis of the cell wall, the bacteria have been divided into two groups, gram-positive bacteria and gram-negative bacteria. So let's see what is this gram-positive and gram-negative, the difference between these gram-positive and gram-negative bacteria. Gram-positive and gram-negative. There was a technique developed by a scientist known as Christian Gram, a technique in which they stain bacteria. When you observe gram-positive bacteria, they visualized that these were purple in color because they were able to retain the stain. But when they visualized the gram-negative bacteria, they were not able to retain the stain, purple stain, and when they were counter-stained it, they acquired the red color of the. So on the basis of this and gram-negative bacteria. In gram-positive bacteria, the cell wall is thick. There is thick peptidoglycan layer about 40 nanometer. But in gram-negative bacteria, the peptidoglycan layer is thin. Thin peptidoglycan layer. And it is approximately 2 nanometer. 2 nanometer in thickness. So this was the structure of the gram-positive and gram-negative bacteria. Let's see some examples of gram-positive bacteria. Example of gram-positive bacteria is Staphylococcus aureus. Staphylococcus aureus. And gram-negative bacteria is E. coli. Now the cell membrane. The next structure we are going to study is the cell membrane of a bacteria. Cell membrane. As you all know that the cell membrane is made up of phospholipids. The cell membrane of bacteria is made up of phospholipids. It has two functional portions. It includes phosphate group and two fatty acids. And these two groups are joined by a glycerol. What is the function of the cell membrane? It helps in transport of molecules. Transport of molecules into and out of the cell. So this was we have studied till the capsule, slime layer, cell wall and cell membrane of a bacteria. Now we are going to study a different type of structure that is present in bacteria. These are the mesosomes. Mesosomes. What are mesosomes? These are the infoldings of the plasma membrane, and these are finger-like and help in cell division. This is the bacterial cell wall, and this is the plasma membrane, suppose. And the mesosomes are invaginations of these plasma membranes. So these are the mesosomes. So just write down what are mesosomes. These are the infolding of plasma membrane. These are finger-like projections that help in cell division. So we have studied cell membrane, cell wall, capsule, slime layer of a bacterial cell wall. So this is in the next class we will be studying about the other structures present in a bacterial cell wall. Thank you.